Hey guys, Antoine Classent from University of Paris in France.
Hi, good morning, everyone, and thank you to the organizers for giving me the opportunity to talk today. And more generally, I would like to thank you, the organizers for selecting so many Vargin-related abstracts this year. Great posters yesterday. Apparently a significant increase of Vargin-related abstracts compared to the previous year I attended this conference. And if you allow me to extrapolate, according to my quick and dirty analysis, by the year 2051, all abstract in this conference will be about Vargins, and I will then retire happy. Now, if you are new to the wonderful world of Vargins, this little summary is for you. However, my group is interested more generally in trying to understand how the parasite is able to evade the immune system in chronic asymptomatic infection for a long period of time. The central dogma of antigenic variation in malaria tells us that the parasite is able to regularly switch its surface antigens in order to escape the immune system. But it's actually very difficult to generate direct evidence for this model. And the two main approaches we've been using so far is from chronic infections from Africa, which we're trying to characterize the parasite transcriptome in collaboration with Ian Cheeseman. But the second approach I'm going to present today is the controlled human malaria infection, so the CHMI that was performed in Gambia a few years ago, in which we selected 19 Gambian volunteers, more specifically 9 serohigh volunteers with relatively high antibody level against 6 falciparum antigens, and 10 serolow with relatively low antibody level against the same antigens. These people were infected with 3-7 again sporozoites and treated as soon as symptoms occurred. And we got a blood sample for measuring Vargin expression by QRT-PCR. Here are the growth curves, with each line being the parastemia in a volunteer. In most serolow individuals, the parastemia grows exponentially. Two interesting exceptions here in purple are infections in which we observed a latency. So the first few days, we don't detect any parasites in the blood. They come up later. And that's exactly what Anna Bachmann and colleague found in Gabon in another CHMI, also with 37, a few years earlier. And they call them controllers because these infections, the host is able to somehow control the infection, and non-controllers. So it's also what we find here. And here is the Vargin expression of the two data sets already combined. Each row is one of the 60 Vargins. Each column is a non-controller from both Gabon or the Gambia. Each orange or red square indicates that this particular Vargin is expressed in this particular individual. The pie chart summarizes the exact same data set. Basically, in non-controllers, a lot of group B Vargins are expressed. However, in controllers, we observe a more restricted repertoire of Vargin being expressed, fewer different types of Vargins. So 3D7 expresses different Vargins based on the host immunity, not on the geographical origin of the host. What I haven't told you yet is that for many individuals, we've got 2 time points, which allows us to record a change in Vargin expression in between the 2 time points. I'm showing just one example here for simplification. We recorded Vargin expression on day 14 and day 20, so 6 days later, in the exact same infection. Again, in orange and red are the expressed Vargin. And as you can see, there is no overlap between the 2 time points. And that surprised me because it means that there is a complete change of Vargin expression between the short period of time. The big question here is it actually did all parasites switch? Or actually only a handful of parasites switched, and most of them, the parasites that did not switch, have been selected against. And the parasites that did manage to switch, they're being selected for. So it's actually very difficult to disentangle between these 2 models. And I thought I could try to disentangle by using culture adaptation, because obviously in a culture flask, there is no immune selection. So we should be able to identify pure switching in vitro. And so we put them in culture for exactly 6 days, because that's the time span between the 2 time points. And the middle row is now the in vitro virgin expression after 6 days. So we first find evidence of parasite that switch off virgins, like these 3 group A virgins here, for example, expressed at day 14. We don't find their expression in vitro nor in vivo, indicating that the parasites have switched away from this virgin and switched toward what virgin? Well, at least this var number 17, because it was not detected in vivo. And then, for whatever reason, the parasite decided to switch away from these group A virgins and then switch on the B17, both in vitro and in vivo. However, we also find an evidence for selection, because this var19 was expressed in vivo, still expressed in vitro 6 days later. However, in vivo, all these parasites expressing that particular virgins have disappeared. So my conclusion is that we find evidence for intrinsic parasite switching and selection in the same individual. Which leads me to this probably very naive model in which 3D7 in non-controller and naive people would first express a large repertoire of group B virgin coming out of the liver. And these parasites can grow exponentially because there is no little immune selection. In controllers, well, I presume that parasites coming out of the liver, they don't know the status of the host. So they would express the exact same group B virgins also in controller. However, because of pre-existing antibodies, we don't detect these parasites because they're being selected against. And this selection would explain the latency. That would explain why for the first few days we don't detect any parasites, because they don't express the non-zero recognized virgins. And it's only when the parasites have managed to switch away and express different virgin that then finally they can grow exponentially. But to test this model, we need the antibody data. And this is what Mark Travassos and colleagues have generated. Just briefly, a PFMP1 is a series of DBL and CIDR domains. You can make recombinant proteins with each of these individual domains, spot it on an array, and then incubate the plasma to determine what antibodies recognize what PFMP1 domain, and they tested the plasma sample before the CHMI, during the CHMI, and after the CHMI. And here is an overall look at the results. In blue, you can see the zero high individuals, which most of them recognize most PFMP1 domains. However, zero low individuals, so individuals with low amount of antibodies in general, also have low amount of antibodies against PFMP1 domains. So it makes complete sense, I think. But what about the dynamics between day 0 and day 35, before and after CHMI? Well, that's what I'm showing right here, is a fold change before versus after the CHMI. In the zero high individuals, actually not many changes. Probably these people have already reached a plateau of the number of PFMP1 domain that they can zero recognize. However, in zero low individual, we do see a significant increase of zero recognition of multiple PFMP1 domains. And perhaps surprisingly, the domain with the highest amount of increase of zero recognition is the ATS domain. And I say surprising because it's the exception to the rule. It's the only domain that is conserved because it's found intracellular. It's located intracellular in the cell. And so clearly, we don't expect these antibody to confer protection, but they are indeed a very good marker of exposure. Now, unfortunately, we didn't have time to finish the analysis of the virgin expression versus the antibody level, but it's clear from what we've done so far is that there is no strong direct correlation between virgin expression and antibody levels. So this nice looking model here is clearly a bad oversimplification of a highly complex parasite-host interaction. And just briefly, we also put all of these as isolates into culture for up to 50 days and recorded virgin expression by qPCR again. Isolates from zero low individuals all converge to the same virgin expression after a few days. It makes sense because there is no immune pressure, so the low express the favorite virgins. However, the isolates from the controllers still express a distinct virgin expression even after 50 days. So if you're interested in that, I will show you the actual data later. And in fact, some of these isolates grow slower compared to the serial low in infection isolates. Last but not least, I would like to thank everyone involved in the CHMI, the volunteers, but also the people at the MRC Gambia and Tön Busman's team, Mark Traversals for the protein array data, my colleague Ovidiu Radulescu for data analysis and supervision, and two excellent PhD students, Inayat and Prince, who generated everything you've seen today, and in fact, it should have been them presenting. So thank you for your attention. I'm happy to take any question, even if it's not about VAR genes.